This is a guide of how you can perform the ELISA test for the 3 isotypes of rheumatoid factor, or RF3. It will give you visually all the information you need to do the test, including the use of the TERRA software. If this is a new unopened kit, please note the content, the expiration date, and the lot number. The lot number is linked in the software and you will notice it later. Please check the lot number in TERRA. If your lot number is not in TERRA, upload the data from the CD. Open the box, take out the reagents, and allow them to warm up to room temperature for 30 to 45 minutes. Set the timer. The kit contains a data sheet that gives the acceptable range of measurements for the calibrators and controls. Take note that the acceptable ranges will change with each new kit lot. The kit information is uploaded in the computer using the CD every time a new lot is provided. If the kit was used before in the lab, that information is already uploaded. Here is a list of all the equipment required to successfully perform this assay. Please check that everything is available on the bench. While the kit reagents are warming up to room temperature, label individual dilution tubes for the IgG calibrator, IgG positive control, IgG negative control, and all patient specimens that will be tested. Place the labeled tubes into your dilution rack as shown on the left side of the image. In order to determine the amount of pepsin buffer and pepsin concentrate required to prepare the pepsin solution for sample dilution, open the software designated RF3 Pepsin Dilutions Calculator. In the box for the number of tubes, enter the number of total dilution tubes needed. Remember to include the calibrator, positive control, negative control, and all patient specimens. In this example, the number is 12. Note the volumes required for the 2 components, which are pepsin buffer and pepsin concentrate. Use the 15 milliliter conical tube to prepare the pepsin solution from 2 components, pepsin buffer and pepsin concentrate. Make sure that the label is for the pepsin buffer and not another buffer in the kit. As provided by the calculator, pipette the amount of pepsin buffer followed by the pepsin concentrate into the dilution tube. Gently invert the 15 ml tube with the pepsin solution 5 times to get a good mix, but do not shake. From the 15 ml tube, pipette 500 microliters of the pepsin solution into all of the dilution tubes designated for the IgG samples. In this step, make sure to use a fresh pipette tip for each tube. Also, move each tube that receives a specimen upward. For example, all the tubes from row F will be moved up to row E. Pipette 5 microliters of the IgG calibrator, IgG positive control, IgG negative control, and patient specimens into their designated dilution tubes. Again, make sure you move each tube to a new row. Firmly cap all dilution tubes. Slowly invert the dilution tube rack 5 times to mix the samples. Watch as the fluid moves up and down through the dilution tubes. Set a timer for 2 hours and 15 minutes, but do not start the timer yet. Place the rack with samples in an incubator set to 37 degrees Celsius. Also place the Pepsin Stop solution into the same incubator. Now, start the timer. Following the 2-hour and 15-minute incubation period, remove the samples and Pepsin Stop reagent from the incubator and uncap the sample dilution tubes using scissors as shown. Discard the caps. Pipette 500 microliters of Pepsin Stop reagent into the dilution tubes. No need to change the tip. After pipetting 500 microliters of Pepsin stop reagent, use a new strip of caps and firmly cap the dilution tubes. Make sure they are not loose. Replace the lid on the rack and slowly invert it 5 times in order to make a uniform mixture. Watch as the fluid moves through the dilution tubes up and down. Prepare the 1X wash buffer. Using a graduated cylinder, add 900 milliliters of deionized water and 100 milliliters of the 10X concentrated wash buffer. Note, you may already have the 1X wash buffer prepared if the kit was used previously. Following the preparation of the 1X wash buffer, take the label from the concentrated 10X wash buffer and place it on the newly prepared buffer. Initial and label the buffer with the date it was created. On the same rack with the IgG specimens, prepare the dilutions of the specimens to measure the IgM and IgA rheumatoid factor. Place a set of dilution tubes in row B in the order shown in the picture from left to right, and label each dilution tube with the name IgA calibrator, IgA positive control, negative control, and the specimen number. This negative control is for both IgM and IgA. In row C, as shown in the picture, label the tubes IgM calibrator and IgM positive control. In a 15 or 50 ml conical tube, pour 1X diluted wash buffer, a little more than the total number of tubes for IgM and IgA on the rack. For example, for 12 tubes, add about 14 milliliters. This will be used as the specimen diluent. From this tube, pipette 1 milliliter of 1X wash buffer with the pipette shown into each of the uncapped dilution tubes for the IgM and IgA calibrators, controls, and specimens. With a new pipette tip for each sample, pipette 5 microliters, not 10, of the IgA calibrator, IgA positive control, IgM calibrator, IgM positive control, negative control, and all of the patient specimens into their designated dilution tubes. Each time a specimen is added, the tube is moved up 1 row. The tubes for IgA are moved from row B to A. The IgM calibrator and positive controls are moved from row D to C. Once again, firmly cap all the dilution tubes. Slowly invert the dilution tube rack 5 times to mix the samples. Watch as the fluid moves up and down through the dilution tubes. The image or map on the left displays an example of the RF3 assay to show where the diluted samples will end up. It shows where you will pipette each of the 3 enzyme conjugates, IgA yellow, IgM blue, and IgG green. Take a moment to review this image. It is an integral step in successfully performing the RF3 assay. If misinterpreted, the assay will fail. In preparation for sample loading, set a timer for 30 minutes. Make sure to load the entire plate within 5 minutes for accurate results. Using a multi-channel pipette, draw 100 microliters of the IgA calibrator, IgA positive control, the negative control, and patient specimens diluted in 1X wash buffer. Pipette the samples into rows A and B of the microplate. These rows are annotated by the yellow arrows presented within this image. Next, move the dilution tubes containing the negative control and patient specimens diluted in 1X wash buffer to the 3rd row of the dilution rack, already containing the IgM calibrator and IgM positive control. Using a multi-channel pipette, draw 100 microliters of the IgM calibrator, IgM positive control, negative control, and patient specimens. Pipette the samples into rows C and D of the microplate wells. Refer to the arrows presented in this image for clarification. With the multi-channel pipette, draw 100 microliters of the IgG calibrator, IgG positive control, IgG negative control, and patient specimens, all diluted in pepsin solution. Pipette the samples into rows E through H of the microplate wells. These 4 rows are annotated within the image of the slide. Set the timer for 30 minutes and press start. When the 30-minute timer sounds, make sure that the strips of wells are firmly placed within the plate frame. This will prevent the wells from falling out of the frame. Hold the frame tight, then carefully flick the microplate into the sink in a swift downward motion. Pour 1x wash buffer into the clean reservoir. Set the multi-step pipette to number 5. At this setting, each tip will load 250 microliters. Insert 8 new pipette tips firmly. With the tips in the wash buffer, push the multi-step pipette plunger down to aspirate the wash solution. Carefully dispense the wash buffer once into each column. Avoid splashing. Empty the wash into the sink with a fast flick of the plate. Repeat this wash procedure 2 more times for each plate. In total, there will be 3 washes per plate. If you still need to use the pipette tips, lay the pipette down on the bench with the tips facing down and out. This will prevent any fluid backing up into the pipette and contaminating it. Firmly and carefully tap each plate upside down onto a paper towel. This is to remove as much excess wash buffer as possible. Set a timer for 30 minutes, but do not start it. Use a new separate reagent reservoir for each of the 3 enzyme conjugates. Pour the IgA, IgM, and IgG conjugates into their designated reagent reservoirs. Use new pipette tips to load each conjugate. With a multi-channel pipette, draw 100 microliters of IgA enzyme conjugate and pipette into rows A and B of the microplate wells. See the yellow arrows for reference. With a new set of tips, aspirate 100 microliters of IgM enzyme conjugate and pipette into rows C through F. See blue arrows for reference. With a new set of tips, aspirate 100 microliters of IgG enzyme conjugate and pipette into rows G through H. See the green arrows for reference. Here is how a fully loaded plate should look after loading the 3 enzyme conjugates. Start the 30-minute timer. During the 30-minute incubation period, all of the leftover conjugate within the reagent reservoirs may be poured back into their respective reagent bottles. Repeat this wash procedure 2 more times for each plate. In total, there will be 3 washes per plate. Firmly and carefully tap each plate upside down onto a paper towel. This is to remove as much excess wash buffer as possible. Use a new reservoir to add the chromogen. Never reuse a reagent reservoir for any solution differing from the original solution in the reservoir. For example, if you have a dedicated reservoir for chromogen, mark it, rinse it, and you can reuse it at a later date. The full plate of wells will require 10 milliliters of chromogen added to the reagent reservoir. If you are running 2 plates of wells, add 20 milliliters of chromogen. Use new pipette tips to dispense the chromogen. Avoid touching the side of the wells. It may cause cross-contamination. Set a timer for 15 minutes and start it as you begin to load 100 microliters of chromogen into the first column of wells. Do not wait until the entire plate is loaded with chromogen before starting the timer. Do not pour chromogen back into the reagent bottle. This will contaminate the rest of the chromogen. Discard any leftover chromogen. Here is an example of a microplate with the chromogen following the 15-minute incubation period. You will see that the calibrators and positive controls are blue. Before the timer stops, pour the stop reagent from its bottle into a new reagent reservoir. Load 100 microliters of stop reagent into the wells after the 15-minute incubation. The stop reagent must be added within 1 minute of the timer ending. Tap the side of the plate gently to equally mix the stop reagent within the wells. The following image is an example of a completed RF3 assay. The calibrator and positive control wells are indicated within the image. The rest of the stop reagent may be poured back into the reagent bottle. Place the microplate into an ELISA reader for analysis. In order to read the microplate using the Terrace computer software, select File, Reading, and New. Select RF3 from the list of assays presented. Next, select Reader, followed by Read and Collect. The reader will take 75 to 85 seconds to read the plate. The following image is an example of the absorbance measurements displayed on the Terrace software for the RF3 ELISA assay. If further clarification is necessary, refer to the slide with the sample layout. After the absorbance measurements and results have been displayed, select the Sample IDs tab as presented within this image. Select Edit On. Enter the patient sample IDs from the assay in the same order the samples were loaded into the microplate wells. After all sample IDs have been entered, select Save. Within the dialog box, type in the initials of the assay, followed by the date and the initials of the individual who ran the assay. An example of this specification is presented within the text of this slide. Select OK to save the assay. This concludes the presentation provided by Theratest Laboratories in reference to performing the RF3 ELISA assay, as well as a demonstration of test result storage within our proprietary software, Terrace. If further clarification or assistance is required, feel free to contact our technical support department at 1-800-441-0771.